Hello, everyone, and welcome to today's Keynote webinar titled Linking Single-Cell Function to Multi-omic Analysis Using Lab-on-a-Particle Technology. This webinar is a part of the 7th annual event in the Laboratory Automation Virtual Events Series. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Dr. Dino Di Carlo, Armond and Elena Hairapetian Chair in Engineering and Medicine, and Professor and Vice Chair of Bioengineering at the University of California, Los Angeles. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Dr. Di Carlo, welcome. You may now begin your presentation.
Thank you so much for the introduction, and it's a pleasure to speak to you all here virtually. I'm going to share some of the work that is really quite new in our lab on using particles as a way to perform single-cell analysis. So many people are, I'm sure, familiar with kind of lab-on-a-chip technology and microfluidics, and that's an area that we've worked a lot in over the last decades. And so we're kind of moving to the next level of how can we use the infrastructure that's in place today, things like flow cytometers, things like single-cell sequencing systems, and by adding a new unit, a particle that can interact with cells and capture things from those cells, how can we open up new capabilities? For example, linking the functions of cells to their transcriptomes or other genomes or epigenetic changes in those cells. So I want to first kind of lay the foundations for why we are interested in developing tools to analyze single cells and their functions. And it really stems from where we see medicine going forward in the future. So if we think about what I call the 3 pillars of medicine, excuse me, molecular therapies, gene therapies, and cellular therapies. So we're all familiar with molecular therapies. This has kind of been the mainstay of the pharmaceutical industry for the last half a century, including small molecule drugs, and maybe more recently, biologics like monoclonal antibodies. And of course, the process of finding new, for example, monoclonal antibodies is a process of analyzing single cells. So whether that's single B cells and baiting their B cell receptors, or in many cases looking at plasma cells that have highly affinity matured antibodies, and they're in that case characterizing the secretions of those plasma cells. So that's one of the mainstays of drug discovery into the future will be in how do we find best new monoclonal antibodies and ways to analyze those single plasma cells is going to be really important. Of course, gene therapies, we've been seeing a lot of excitement from the new approaches that we have to engineer the genome, CRISPR-based approaches, and many variants of that have come after, as well as new approaches to deliver our packages to cells, so viral delivery approaches and others. So in characterizing functions of cells, one thing that is really important, and we see this in the gene therapy field, where the lack of good ways to make lots of virus is a bottleneck to that field moving forward. And so you can imagine approaches to characterize what makes a cell a good producer of a virus, what transcriptomic or other genetic changes in that cell make it a good producer where it can secrete and release lots of virus. That would be a really important application. And then finally, cell therapies. This has been an area that we've seen a number of FDA approvals over the last 5 to 6 years in the chimeric antigen receptor T cell therapy, where we have receptors that are added to the T cell to recognize antigens that are not presented by by MHC molecules. And those therapeutics are extremely exciting because in essence, they're living drugs, right? They can recognize a target cell that you would want to kill, but also they can release other molecules. They can kill that cell, but they can also release other molecules, cytokines, to attract other cells into the tumor site. Or they can also proliferate, right? And so kind of keeping a sentinel of other cells in case the cancer recurs. Of course, so characterizing the functions of those cells, what they secrete in particular, that has outsized impact on the behavior and how those therapeutics can work. And there's new areas where we're seeing a lot of interest in creating cells that secrete drugs, right? So whether it's secreting monoclonal antibodies or whether it's secreting other proteins that are missing that are therapeutically important for a patient. So we see, that this fundamentally this landscape of therapeutics into the future over the next 40 to 50 years will be dominated by approaches to characterize the functions of cells. And so why is this different than what we had previously, and why do we need new tools? Well, if you think about it, previously when we had small molecules and even proteins, the structure defines the function, right? So the structure of the small molecule, if you can analyze it, NMR or some other approach, then you know that function. If you've characterized that function previously, if it has that structure, it should have that function. It's the same with proteins in most cases, right? You have a protein, you have the amino acid sequence, it folds in a particular way. You should know what if you know that amino acid sequence, you know what the function of the product is if you characterize it. This is not the case with cell therapies. So with cell therapies, even if you introduce a gene, a genetic modification to the cell, that does not necessarily mean that all the cells that have that genetic modification are going to behave the same. Some cells will produce more of that protein. Some cells will produce less. Some cells may secrete more or secrete less. And this all depends on a number of factors. One, epigenetic modifications in those cells. Other signaling pathways that are activated. So the environment and where those cells have been over time. And so this is why it's critical, we think, to have tools to quantitatively characterize functions of cells and also be able to sort and find subpopulations of those cells based on function. And what I'll talk about today is linking that function. If we can analyze and sort based on function, can we link that function to other aspects of the cell, like transcriptomic information? And then we can start to get insights into, okay, well, maybe if we added a set of genes in the signaling pathway or other genes, then that could better define the function of those cells in the future. So this is really important. And so, we think that these new tools that enable us to go beyond what flow cytometers can do now, go beyond what single cell sequencing can do now, and have that functional link, that's going to be critical moving forward. And so what are the challenges to developing tools to do this? One is that the scale of biology is small compared to the scale in which we do, in which we have vessels to kind of assay cells. Excuse me. So the automation approaches for performing biological research is focused on multi-well plate formats, which even if the smallest multi-well plates, wells in those well plates, 1536, let's say wells, that they still have thousands of times larger volumes compared to the single cell. Why is that important? Well, if you're trying to look at what that individual cell is doing, what molecules it's releasing, that gets diluted out in this huge volume, and now you have a very low sensitivity assay, or you have to cells, and now you've lost single cell sensitivity. So we need to try to match the volume scale on which we analyze cells to the scale of the biology. And that's where microfluidics tools have played a role. But what we've seen over the years is that being able to adopt new microfluidics tools has been a bottleneck, right? We have a lot of tools like flow cytometers that are in labs already, but we don't have new microfluidics tools, especially if we need a new instrument for each niche application. So our approach was, can we leverage things like the flow cytometers, things like single cell sequencing systems, like 10x chromium systems or other systems, in order to perform assays on the function of single cells. And this is where this lab on a particle concept comes from. And in particular, we have a type of lab on a particle that we call a nanovial. So a nanovial is a hydrogel particle that holds a volume that's less than a nanoliter. You can have thousands or millions of these in a tube. This is what they look like. I would encourage the audience to click on this video to play it. These are nanovials that are settling down into a well. And so you can just see the structure of them and how uniform they are. So they have these, it's basically a little bowl that has a little cavity in a hydrogel particle. And that cavity is suited to hold cells. You can make them in very uniform ways, and we actually use microfluidics to make them. So what do these nanovials do? Well, they create uniform compartments to hold the cells and capture their secretions. They have that solid substrate so that you can perform reactions. So you can capture the things that the cells are secreting. You can add reagents to label what is being secreted. And then they have the ability to analyze and be sorted in high throughput using traditional cytometers. So the hydrogel itself can be functionalized. So it can be functionalized to have cell adhesive moieties, can be functionalized to have antibodies to capture secretions from cells that can then be labeled. And this is what it looks like. In one case, you have cells here in pink. Their secretions, in this case of a monoclonal antibody, are captured on the cavity here, and then they're labeled with a fluorescent secondary antibody. And they're compatible. They can be run through the cytometry systems, in this case showing the Sony sorter, which we use often. So just want to show you kind of a general workflow for how to use nanovials, or how we've been using nanovials and others are using them. One is you basically load cells by mixing nanovials into a volume of, mixing a volume of cells into nanovials. So they are randomly encapsulated and captured based on adhesive moieties, for example, a cell capture antibody to a cell surface protein. You then incubate. You can remove unbound cells. You incubate these so that the cells then secrete something of interest to characterize their function. You can capture what they're being secreted by also having capture antibodies on the surface. You then wash and then label. So in this case, you can label with either fluorescent and or oligotagged antibodies. And so I'll talk more in this talk, especially about our work on linking that functional information about secretion with this transcriptomic information where the oligotagged antibody is useful. Again, these can be sorted, they can be grown, cells can be grown up again, they remain viable. Actually, the nanovials protect the cells from shear stress. The cells can be expanded, or they can be analyzed. Their secretions or protein markers can be analyzed. How do we fabricate them? As I mentioned, we use microfluidics to fabricate them. We have a PEG polymer precursor that it can be photo cross-linked. Then we have the cross-linker components and dextran in another phase. We co-flow these. We form a droplet. We allow this droplet of PEG and dextran to phase separate to form a PEG-rich and a dextran-rich phase in oil. And then we cross-link that with UV light, and we remove the dextran that was not cross-linked to create these bowl-shaped nanovials. So that allows us to make a lot of these nanovials very easily. So we are calling this microfluidics 2.0, and that you're using microfluidics initially to make these particles, but then the end user or biologist or engineer or chemist, etc., they are using the nanovials directly as their microfluidic compartments. And so that enables a really easy process for the end user to use the standard instruments and standard protocols without having any knowledge of the microfluidics. This is relatively new, and these are some of the papers that we've been publishing in preprints, applying this technology for antibody discovery, applying it for characterizing, defining a functional repertoire of T cell receptors based on cytokine secretion when an antigen antigen-specific T cell binds to a nanovial. But today I'm going to talk mostly about our work on what I call SEC-seq, or secretion-encoded single-cell sequencing. And this is where we are linking the information of secretion from a cell. We're linking that to the transcriptome of that cell. So we can actually have functional information annotated on top of the transcriptome. And I'll talk about some of our work using that for looking at the secretions of growth factors from mesenchymal stromal cells, and some of our collaborative work with Richard James' lab led by Renee Chang on correlating the antibody secretion of plasma cells to the transcriptomes that lead to high secretion. So the first kind of just step in any of our assays is cell loading. So how do we load cells on these nanovials? Well, the key step of loading is functionalizing the nanovials with cell adhesive moieties. In this case, I'm showing a few different examples of cell adhesive moieties. One could be antibodies against cell surface markers, could be anti-CD138 for plasma cells. It can also be antigens or peptide MHC complexes to bind specific antigen-specific T cells. We have a formulation where we can put gelatin just in the cavity here. So extracellular matrix components that allow, for example, adherent cells like mesenchymal stem cells to adhere, or CHO cells that produce high levels of antibodies that we want to use in the production of these monoclonals. So let me introduce this concept of secretion-encoded single-cell sequencing, or SEC-seq. This is led by my student along with Shreya Udani, along with Justin Langerman from Catherine Plass' lab here at UCLA. And they've done an amazing job to develop this new approach to link transcriptomes and secretions from single cells. So how does this workflow work? Again, you start with a tube of nanovials. You load your cells onto the nanovial. There's captured antibodies for their secretions of those cells. We allow them to secrete. We then label their secretions with an oligobar-coded detection antibody. This whole entity, the cell, along with its secretions that are oligo-tagged, goes into the droplet of a 10x chromium system for doing single-cell transcriptomics. And here then we basically now get information of the transcriptome of that cell, as well as the bar-coded information about what that secretion was from that cell. So again, look, you can get information, for example, from the transcriptome. You can get the UMAP or some type of dimensionally reduced transcriptomic information where you find clusters of cells. And then you can overlay on that. Okay, well, where was the oligobarcode associated with secretion, and what is unique about those cells in their transcriptome that should correlate with that secretion? So in this case, we're focusing, excuse me. In this case, we're focusing on mesenchymal stem cells, or mesenchymal stromal cells. And one therapeutically important growth factor, VEGF, that's important for cell therapy type applications where folks are interested in using these cells to promote regeneration of tissue when injecting into patients. So the first question is, can we get an assay like this to work? Are nanovials compatible with cell transcriptomic workflow? Many questions we had. And the short answer is yes, we can get the assay to work. This is just showing some data on VEGF dose response. So how does the assay work on the nanovial? As we increase the concentration of recombinant VEGF, what kind of signal increase do we see on the nanovial? And we can see about more than 2 orders of magnitude increase in signal, and so spanning that range of signal for the nanovial. And then when we add cells that allow them to secrete, we see an increase in signal of the anti-VEGF antibody over time as they secrete more. And without capture antibody, you don't see that signal. And also what we see, the signal is different for cells that are exposed to hypoxic versus normoxic environments, so which should trigger different amounts of VEGF secretion. And so we see that as well. And importantly, suspended cells actually had less viability after sorting in this workflow than cells on nanovial. There's some protective effect to being on a nanovial. We then looked at whether nanovials can be introduced into these emulsions without leading to any problems there. We can see that yes, you can encapsulate the nanovial. Here's a gel bead from one of the test kits. So you can see the MSC on the nanovial next to the gel bead, all in the emulsion. When you have the lysis reagents, you can see that calcinium labeled cells that they get lysed upon being entered into the emulsion, but that doesn't spread out over many droplets, so it's localized. And we can control the number of nanovials in droplets in a similar way that you can control cells following Poisson statistics. We did initial mixing experiments where we had human cells and mouse cells on nanovials, and we didn't see, we saw that they mixed as expected. There was no extra mixing. Something slightly interesting here is that the transcripts per cell that we recovered with nanovials is slightly higher than unsorted cells or sorted cells. We also did a mixing experiment where we barcoded the nanovial type. So we had 2 different treatments, a normoxic treatment and a hypoxic treatment. And what we see is, yes, we can kind of pull out the normoxic barcodes from the hypoxic barcodes on the nanovials. And if you start to look at the mixed UMAP and the barcodes overlaid on that, you can see the hypoxia signal, transcriptomic signal, overlays very nicely with the hypoxic barcode. So this is giving us confidence that this type of workflow works. We then did a full SEC-seq workflow. So here we're looking at gene expression and then also the VEGF secretion. We did this with a combination of normoxic and hypoxic cells together. So the first thing you see, so this is the UMAP of the transcriptome, and then overlaid on that, here's the secretion. So you can see that we had some expectation that hypoxic would have higher secretion. You see that both with the fluorescent signal here as well as the barcode signal. So on average, the hypoxic cells have higher secretion signal, but it's not uniformly distributed. So this is really exciting, right? Because it's telling us this functional information is linked to some subpopulation of cells that have higher levels of secretion. So then we wanted to explore that further. So we started to explore this a bit further. So first I want you to look at the transcripts here. This is the log, the VEGF transcripts levels. So there's definitely higher transcripts are induced with hypoxia. Now, if you overlay that with secretion, they do not overlay, right? So higher transcripts does not mean higher secretion of VEGF in this case, right? So you can see higher transcripts in this region, but higher secretion is in some of these subpopulations like M1 and M2. So this is very exciting because this allows us to understand that there's some subpopulations that are transcriptionally defined that have higher levels of secretion. So you can look at these different clusters. I'll generally put your attention that the transcripts in the normoxic ones, N1 through N5, are lower than the hypoxic ones, H1 through H4. But if you look at M1 and M2, which are mixed hypoxic and normoxic that have high levels of secretion, those ones have different levels of transcripts, right? So there's both have high levels of secretion, but different levels of transcripts. And in fact, if you look at the top right graphs, you see that the normoxic cells in particular, there's basically no correlation between transcript levels and the amount of secretion of VEGF. For hypoxic cells, you see that it's slightly correlated, but not very strongly. In fact, if you look at the bottom right graphs, you can see that some of the most highly correlated gene expression to VEGF secretion are other genes that are not related to VEGF, right? So these insulin-like growth factor binding proteins, IGF-BP6, IGF-BP5, other proteins related to extracellular matrix like fibronectin, et cetera. So this is quite unexpected for us and interesting. So again, here's some of the highly correlated genes, and this is across both the normoxic and the hypoxic cells. You can see that there are a subset of highly correlated genes in these clusters, like these insulin-like growth factor binding proteins. IGF-BP5 very well defines that cluster, its transcripts. And so that's really an interesting, understanding why those genes start to do is really important to if you want to control VEGF secretion. We also repeated this experiment. This is just another set of normoxic cells. And again, we found this high VEGF secreting population. This is showing VEGF barcode. Again, the subpopulation here is high VEGF secretion, and it also correlates with these specific transcripts. So this very repeatable result. And again, actually, we can show this across a number of different experiments where we always have a population about 5 to 20% of the population that has this high secretion phenotype. And this high secretion phenotype has some consensus genes associated with cell motility, response to wounding, blood vessel development. And so we actually define a new gene signature, which we call the vascular regenerative gene signature. And that's what we're plotting here, that this subpopulation of vascular regenerative gene signature is about 5 to 20% of MSC population. And those are the cells that are largely responsible for secreting most of the VEGF, especially in normoxic cells, and then in hypoxic cells, you have some other cells that also secrete VEGF. So that's very interesting and important. And some of the conclusions, you think about this, if you start to, so yes, I think some of the conclusions here that are really important is that if you wanted a cell to produce high levels of a gene product and secrete it, just controlling the gene expression is not sufficient. This is what this data is saying, is that may not be sufficient to just control the gene expression, that you also need to control other pathways maybe involved in secreting and the energy required to create the molecule, create the proteins, et cetera. I want to then kind of just give another example of using SEC-seq. This is led by our collaborators at Seattle Children's Hospital, Renee Chang and Rich James in his lab. And so the experiment here is very similar in some ways, but very different in that we're using human B cells that are differentiated into antibody secreting cells in vitro. Those are then loaded into nanovials. We capture them based on surface markers and allow them to secrete IgG. We then can perform assays to characterize their secretion of IgG, imaging flow cytometry, for example, with the image stream, other flow cytometry approaches, or also barcoding and downstream single cell sequencing, so the SEC-seq workflow. But basically differentiating B cells, loading them, allowing them to secrete, and then sorting out the viable cells with the NanoSelect system here, and then using the 10x system for the SEC-seq workflow to characterize the barcoded IgG that's secreted, as well as the transcriptomes of those plasma cells. Here are some of the data from these experiments, and this is looking at data from 3 donors one donor that was an earlier state of differentiation, day 10, and then 2 donors from day 13. And so here's the secretion encoded cell information, so higher IgG barcodes. Here's the transcriptomic UMAPs showing the subpopulations. You can see that the day earlier in differentiation that you have more of this PAX5 transcription factor. So actually, in fact, the majority of this cluster 6 here is from the early, less differentiated cells. And then these bottom ones are associated with higher, more differentiation, more transcription factors with plasma cells. And you can start to see that if you start to look at the IgG secreting cells, that aligns very well with the IgG secretors. So you can look at transcripts for IgM and IgA and then double negative, and then also look at IgHG genes, and then look at the heavy chain genes, and those are all kind of in these bottom parts of the UMAP. And then we can also characterize the high seq IgG high, so high levels of the barcode, which we're assuming are the high secretors of IgG. And so what we find is that we see that the seq IgG high are with the clusters that have high level are the IgG secretors. That makes sense. So the red ones here are all the high secretors. Percentages here, and if you go to the ones that are IgM or IgA, there's very low levels of this barcode. And in particular, there's some interesting thing here where number 10, which is one of these subpopulations, has even higher IgG secretion. You can do some pseudotime analysis, and you start to see that as pseudotime increases, as the cells become more differentiated to plasma cells, the number of IgG barcodes increases, so that they secrete more. And then you can start to analyze what are some of the genes that are correlated with this high IgG production, and maybe identify new markers for plasma cells. So an example here is markers for example, CD59. So CD38 and CD138 are kind of known plasma cell markers, but CD59 is a new marker that comes up in our highly correlated, as well as differentially having high seq IgG barcodes. And so those CD59 shows up nicely as being highly expressed in those cells that have high levels of secretion of IgG. And then we can go back and test that. So even though it's expressing that gene, is it actually producing it on the cell surface? And so yes, what we find is that if you isolate the cells based on the high levels of CD59, that those cells on average will produce more IgG. And that, in fact, as much as good of a marker as CD138, really. And there's many things that we found. So besides kind of new surface markers, we also find some other mechanisms that play a role in this high IgG secretion, including energy production. So what we found is a lot of genes involved in energy production. And then if we go back and stay in the mito-high versus mito-low cells, they also have differences in IgG secretion, or endoplasmic reticulum. So a lot of genes involved in endoplasmic reticulum, protein folding, dealing with a lot of oxidative stress, really in genes that are required if you have lots of proteins being made really rapidly. Then we see high levels of secretion associated with those genes. So this is exciting. It kind of lays a new foundation for, let's say now, engineering cells that have high levels, that can produce higher levels based on the transcripts that they produce. So to conclude, we are excited by this new approach to characterize the function of cells and being able to link that to underlying transcripts and surface proteins. And so the cell analysis we think is incomplete. We need that linkage to function in order to really understand what all of this information means that we're getting from transcriptomes. Because in the end, it's the function of the cells in vivo that's driving the therapeutic effects, especially in the cell therapy fields that we're interested in kind of improving. And so nanovial technology, that lays the foundation for linking function to these traditional biomarkers, and we think will help refine cell identity. And as I said, engineered cell therapies can be better defined if we start to have functional approaches to characterize them, and then go back and say, okay, based on these functional approaches, how do I engineer the genome and engineer the epigenome in order to make the cells have these functions? So we see that as an iterative process. So with that, I'd like to thank you again for listening, acknowledge some of the key contributors, my students, Shreya Udani, and Justin Lingerman from the POTH Lab, our collaborators at Seattle Children's, Richard James and Renee Chang, also Joe Doretti, who is one of the initial inventors of this nanovial technology, and is now leading a company called Partilian Bioscience, which is commercializing nanovials and making those available for others to perform similar types of assays as we are doing it in our lab. With that, thank you so much.
Thank you, Dr. Di Carlo, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Now let's get started. Our first question here is, what types of cells are nanovials compatible with?
Yeah, that's a great question. So far we've used a number of different cell types. As I mentioned in today's talk, we've used like plasma cells, we've used mesenchymal stromal cells or MSCs. We've also used T cells. We've used hybridoma cells, CHO cells, fibroblasts, macrophages. Yeah, a number of different cell types. It's really limited by the cell capture moieties that you use on the nanovials.
Thank you for that. Our next question is, how do you load individual cells into nanovials?
Yeah, so it may look like there are always single cells in nanovials, and that's after sorting. We sort the ones that have single cells, but in the process of loading, it follows a random distribution following Poisson statistics. This is similar for any like droplet microfluidic approach, where you load or even limiting dilution, cloning or other things, where you load at a level that is below the number of compartments. So we're usually targeting about 10% to 20% of the compartments. And so on average, we get a single cell in each compartment. In some cases, we've designed the nanovials to have sizes of the compartments so that only a single cell can fit, in which case we can get even higher levels of single cell loading.
Thank you for that. Another question here, for how long can you grow cells within the nanovials?
Yeah, so we've, in the work I showed today, we were growing mesenchymal stem cells up to 24 hours in the nanovials. Remember, these are not fully encapsulated, so there's still media exchange that can go into and out of the nanovial to allow the cells to get nutrients, allow waste to be removed. It's kind of like a little test tube or petri dish for a single cell. So we've grown between 3 hours up to 24 hours. If you start to go to longer time periods, then the cells start to divide and grow out of the cells, grow out of the nanovials. So we're usually limited to about 24 hours maximum.
Thank you. And then someone is asking, what types of calibration do you use?
Yeah, that's a great question. So So normally when we are running assays in flow cytometry, we'll have calibrations for each of the fluorophores that we're using. So we'll have if we're doing like multiplexed assays, we're looking at cell surface markers and secretions. We'll have individual fluorophores that we do individual calibrations on to do compensation measurements. We also, the other thing that requires some calibration for some of the flow sorters is something called drop delay, because nanovials are slightly bigger than most cells, and the drop delay setting is not optimized, and the cell drop delay setting is not optimized for nanovials. We find that for like the smaller nanovials and like systems like the Sony system, that we don't need any drop delay calibration. But as we go to larger nanovials, then we do some calibration to make sure that the delay of the drop delay is calibrated to maximize the sort yield.
Thank you for that. And thank you, Dr. Di Carlo. Do you have any final comments for our audience?
Yeah, thank you so much for your time today, and hopefully this is something that could help your research, and that's one of the reasons we like to bring these share these technologies and bring them out, is so that others can benefit from them, we hope, if there's any interest in doing assays, please let me know.
Thank you again, Dr. Di Carlo, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we didn't have time for or those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until March 22nd, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.